Hello everyone, this is our fifth chapter of Level 1, Mendel's Law, Laws of Inheritance and its Exception, Genetic Disorders and Types. Genetics is the branch of science that deals with the study of heredity and genes. Pythagoras, Aristotle, Hippocrates, Epicurus and others have contributed on the field science, the classical period. Modern genetics was pioneered by Gregor John Mendel. He worked on pea plant. His work was published in 1866. This established the theory of Mendelian inheritance. Inheritance is the process by which characters are passed on from parent to progeny. Heredity is based on inheritance. Variation is the degree to which progeny differs from parents. Variation can be in the sectors of morphology, physiology, cytology and behavioristic traits. This slide is very important. In 1865, Mendel documents pattern of heredity in pea plant. In 1869, Mischer first identifies DNA. In 1902, Sutton and Boveri proposed chromosome theory of heredity. In 1915, T. H. Morgan worked on Drosophila melanogaster and confirmed the chromosome theory of heredity. Then Muller in 1927 shows that X-ray induced mutations. In 1928, Griffith transformation experiment transformed non-pathogenic bacterial strains to pathogenic. In 1930s, Hammerling shows that the hereditary information is contained in the nuclei of eukaryotic cells. In 1931, Matt Clinton demonstrated genetic recombination in corn. In 1941, Beadle and Tatum described the one gene, one enzyme hypothesis. In 1944, Avery, MacLeod and McCarthy shows that DNA is the transforming principle responsible for heredity. 1950, Chargaff discovered that adenine combined with thymine and cytosine combined with guanine, but adenine combined with uracil in place of thymine. This rule is called Chargaff's rule. In 1952, Hershey and Chase used radioactive labeling to prove that DNA is responsible for heredity. In 1953, Watson and Crick proposed the double helix structure of DNA. In 1961, Jacob and Monod proposed the existence of messenger RNA. In 1990s, genome sequencing projects began. So this slide is very important. So Mendel has a great interest in plant hybridization from the start because his father was interested in the same field. Mendel conducted his historic experiment with garden pea in the monastery garden for about 9 years from 1856 to 1864 and published the result in a less known journal in 1865. Plant selected by Mendel is Pisum sativum. Mendel used several contrasting characters for garden peas which are represented below. These are the traits: seed shape, seed color, pod shape, pod color, flower location and plant size. Mendel selected garden pea as material in his experiment due to the following advantages. Pea plants are easy to grow and interbreed. Peas reproduce and grow to maturity in a season, an annual plant. Several generations can be produced within a short period due to short life cycle. The flowers are bisexual, containing both female and male parts. They are self-fertilizing in nature. Several contrasting characters are present in pea plants. It is easy to hybridize because pollen from one plant can be introduced into the stigma of another plant by removing anther. Anther removing is a process. This is also known as emasculation. Mendel's law. Mendel first performed a monohybrid cross in which he crossed tall and dwarf pea plant and collected all the seeds obtained from this cross. He grew all the seeds to produce plants of the first hybrid generation called the F1 generation. He observed that all the plants are tall. Mendel self-pollinated the F1 plants and discovered that in the F2 generation, some plants are also dwarf. The proportion of dwarf plant is 1/4 and that of tall plant is third part of the fourth. So parental generation, tall plant crossed with dwarf, and after he got capital T, small t. Capital T means this is dominant character, small t means this is recessive character, and after this cross he got the ratio of 3:1. Law of dominance. It states that characters are controlled by discrete units called factors. Factors always occur in pairs. In a dissimilar pair of factors, one member of the pair dominates the other member. Law of segregation. The alleles do not blend and both the characters are recovered during gamete formation as in F2 generation. Law of independent assortment. The law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. Two new combinations, round green and wrinkled yellow, are formed in the dihybrid cross due to an independent assortment of traits for seed shape, round, wrinkled, and seed color, yellow and green. This is the checkerboard. Yellow and round color crossed with green and wrinkled. This is known as dihybrid cross, and after this cross Mendel got 9:3:3:1 ratio. 9:3:3:1 is the phenotypic ratio. So points to remember: dominance and recessive. Dominance means when a factor expresses itself in the presence or absence of its dominant factor, called dominance. Recessive, it can only express itself in the absence of or its recessive factor allele. Genes that code for a pair of contrasting traits or characters are known as alleles. So, exception of Mendel's law. Mendel's law too had some exceptions. Here is the list. Incomplete dominance. It is a post-Mendelian discovery. It is the phenomenon of neither of the two alleles being dominant, so that expression in the hybrid is a fine mixture or intermediate between the expressions of two alleles. In Snapdragon mirabilis jalapa, there are two types of pure breeding plants, white flowered and red flowered. On crossing the two F1 plant, it possesses pink flowers. On selfing them, the F2 generation has one red, two pink, one white. The pink flowers are due to incomplete dominance. This is the chart of incomplete dominance. 1:2:1. Next is codominance. It is the phenomenon of two alleles lacking dominance, recessive relationship, and both expressing themselves in the organism. Human beings' ABO blood grouping are controlled by gene. The gene comprises three alleles, IA, IB, and I. Any individual contains any two of three alleles, IA, IB are dominant over small I. The plasma membrane of RBC has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. When IA and IB are present together, Both express their types of sugars due to codominance. Multiple alleles. They are multiple forms of a gene that occur on the same gene locus distributed in different organisms in the gene pool, with an organism carrying only two alleles and a gamete containing only one allele. For example, ABA, ABO blood grouping. So genetic disorders. Genetic disorders may be grouped into two categories: chromosomal disorders and Mendelian disorders. So difference between both these disorders are as follows. Mendelian, these are due to alteration in a single gene, but chromosomal disorder, these are caused due to absence or excess of one or more chromosome. Next, Mendelian, they are transmitted into generation through Mendelian principles of inheritance. In chromosomal, they may be recessive or dominant. And last one is, example for Mendelian disorder are color blindness, phenylketonuria, and chromosomal Down syndrome, Turner syndrome. This is the chromosomal disorders. Aneuploidy, failure of segregation of chromatids during cell division results in loss or gain of the chromosome. Polyploidy, the failure of cytokinesis leads to two sets of chromosome. Down syndrome, it is due to an additional copy of chromosome number 21. The affected individual is short-statured with a small rounded head, partially opened mouth and furrowed tongue. Mental development is retarded. Next is Klinefelter syndrome. It is due to the presence of an additional copy of the X chromosome, XXY. Such persons have overall masculine development with feminine development, like development of breast, is also expressed. Next is Turner syndrome. It is caused due to the absence of one of the X chromosome, 45 with X0. Such females are sterile and their ovaries are rudimentary. They lack secondary sexual characters. Mendelian disorders. Hemophilia. It is a sex-linked recessive disease in which a minor cut leads to non-stop bleeding in an infected individual. It can be transmitted from a heterozygous female to her son. The probability of a female becoming hemophilic is highly rare, since the mother of such a female has to be at least carrier and the father should be hemophilic, unviable in the later stage of life. Next is sickle cell anemia. It is an autosomal linked recessive trait in which mutant hemoglobin molecules undergo polymerization under low oxygen tension, causing the change in the shape of the RBC from biconvex disc to an elongated sickle-like structure. Substitution of glutamic acid by valine at the 6th position of the beta globin chain of the hemoglobin molecule causes the defect. The substitution of amino acid in the globin protein results due to the single base substitution at the 6th codon of the beta globin gene from GAG to GUG. Phenylketonuria. It is an autosomal recessive trait. The affected individual lacks an enzyme that converts the amino acid phenylalanine to tyrosine. Due to this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives that result in mental retardation of the individual. Reduced synthesis of one of the globin chain can cause the formation of abnormal hemoglobin molecule, hence causing anemia. Next is color blindness. It is a condition in which certain colors cannot be distinguished due to the lack of one or more color absorbing pigments in the cone cells of the retina. In humans, the most common color blindness is red-green color blindness, which is a sex-linked defect caused by a recessive gene, and hence more common in males than females. Thalassemia. It is an inherited autosomal codominant blood disease. The genetic defect results in a reduced rate of synthesis of one of the globin chain that make up hemoglobin. Reduced synthesis of one of the globin chain can cause the production of abnormal hemoglobin molecules, thus causing anemia. Points to remember: phenotype is a morphological expression of a single character. For example, tallness or shortness represent the phenotype of the plant. Genotype is the genetic makeup of a cell, an organism or an individual. The specific allele makeup of the individual. Alleles combine to make a genotype, for example, capital TT or capital T small t or small tt. So last one is, these are the questions you have to solve. What is inheritance in genetics? What is the variation in genetics? What are Mendelian disorder in genetics? What are chromosomal disorders? What is equational division in genetics? Who is the father of genetics? So we will meet tomorrow in our next lecture.